Hello, my name is Dr. Philip Mease, Director of Rheumatology Research at Swedish Providence Medical Center in Seattle, Washington. I have just returned from the EULAR meeting in Barcelona, where there was a heck of a lot of interesting abstracts being presented orally and in poster form on a variety of different subject areas in rheumatic disease. I had a special interest in learning about IL-17 inhibition in psoriatic arthritis and axial spondyloarthritis. And as you know, we have available to us two IL-17A inhibitors, secukinumab and ixekizumab. We know that IL-17A is a key pro-inflammatory cytokine in PsA and axSpA. There's also now increasing evidence that inhibition of IL-17F, in addition to IL-17A, can be beneficial. And we've certainly seen data on bimekizumab, an IL-17A and F inhibitor at this meeting, out to 3 years in both PsA and axSpA. There are some new IL-17 inhibitors in the pipeline, and I wanted to make a few comments about them and thinking about where they might fit into the treatment paradigm with three currently available to us. One of them is known as sonelokimab, which is an IL-17A and F inhibitor, and it has a unique molecular construct. It is a nanobody which is molecularly constructed from antibodies of the camel family. So it's a very small kilodalton molecule. And one of the things that we've seen in, for example, treatment of cancers with nanobodies is that they have the capability of getting into difficult to treat tissues like complex cancers. So this is an exploration with a molecule that has a similar construct as other nanobodies, and we're looking at week 24 data from a phase 2 trial with this particular compound. There were 3 doses, 60 milligrams with and without load, and a 120 milligram dose versus placebo. And what we're looking at in a presentation by Ian McInnes is high threshold responses. So we saw minimal disease activity achievement of 60% in the 60 milligram dose, and 120 milligram dose yielded slightly over 50%. These are high responses for this particular composite measure that includes 7 different items with thresholds. So for example, if you get to a swollen joint count of less than or equal to 1, or a tender joint count of less than or equal to 1, or an enthesitis count of less than or equal to 1, and so on. If you get 5 of the 7, you're in a state of minimal disease activity, which we know is a great target for treatment and is correlated with all kinds of good things like improvement of function and quality of life and likelihood of inhibition of structural damage progression. Other high threshold responses included an ACR 70 and 100, which were seen in 35% of the 60 milligrams with load and 48% of the 120 milligram dose. ACR 70 response in 39 and 42% respectively, and PASI 100 in 62 and 63% respectively. So this is clearly an efficacious molecule. It's now proceeding into phase 3. And one of the things that we know about our use of biologics is that even when a patient has been tried on a previous IL-17 inhibitor, there may be benefit from trying a different molecular construct, even though the same overall mechanism And so, especially with the A and F inhibition, we're looking forward to this moving along in both PSA and eventually in axSpA. We also saw some reporting on two other new IL-17 constructs. One was an IL-17A inhibitor from China that was studied in China in patients with radiographic axial SpA or ankylosing spondylitis. With this particular medicine, there was similar efficacy and safety profile as we've seen with other IL-17A inhibitors such as secukinumab and ixekizumab. Don't know whether or not this is going to come out of China and to the rest of the world. We'll stay tuned for that. And then finally, there was a presentation on a new molecular approach to IL-17A and F inhibition in a study known as ORCA-002. In this particular construct, the molecule is modified so that it may have an extended half-life. And in the in vivo and in vitro studies that have been done with this, the molecule appears to have efficacy similar to bimekizumab, which is an approved and now being used IL-17A and F inhibitor. When the drug was analyzed in monkeys regarding half-life and then extrapolated to humans, it was suggestive that the effects can endure for 3 to 6 months, so allowing for less frequent administration. Whether or not these medicines will ultimately make it, we don't fully know yet, but there's promise. And what we've learned over the years, as, for example, when we have gone from one TNF inhibitor to another, is that there can be value in the potential effectiveness of such drugs, even when patients have had an inadequate response to these mechanisms previously. So all in all, both for PSA and axSpA, we are glad to have these IL-17 inhibitors available to us, working effectively to achieve such goals as minimal disease activity. Thanks very much. And for more details and highlights and insights about the EULAR meeting, head on over to Room Now.